Neurodevelopment and genetics in schizophrenia. Now, what seems a very obvious question now is what causes schizophrenia? Nature, that is genetics, or nurture, that is the environmental or epigenetics? The answer seems to be both. It seems more likely that schizophrenia is a conspiracy among many genes and many environmental stresses to cause abnormal development of brain connections throughout life. Not surprisingly, there is no single gene for any specific psychiatric symptoms, behaviors, personalities, or temperament. Today, mental illnesses are thought to be linked in part to someone inheriting an entire portfolio of many genes that carry risk of mental illnesses. Multiple risk genes, any one of which alone may be clinically silent until stress from environments puts a load on these defective genes. And these can be any kind of stress, traumatic experiences, learning experiences, sensory experiences, sleep deprivation, toxins, and even drugs. And these are all examples of how normal genes, such as those that regulate the information and removal of synapses, are turned on or off by environmental stresses. Cannabis seems to be particularly malicious environmental stressor to those vulnerable to schizophrenia. These are all examples of the notion of experience-dependent development of synaptic connections, something that is hypothetically abnormal in schizophrenia. Best evidence for the role of disconnectivity of genes in schizophrenia is multiple genes that regulate not only neuronal connectivity in general, but glutamate synapse formation and removal in particular. These genes include disbindin, neuroreglin, ERBB4, and DISC1. Disbindin is also known as dystrobrevin binding protein 1 and is involved in the formation of synaptic structures and regulation of activity of the vesicular transporter for glutamate, that is VGLUT. Neuroreglin, on the other hand, is involved in neuronal migration, genesis of glial cells, and subsequent myelination of neurons by glia. They also activates an ERBB4 signaling system that is co-localized with the NMDA receptors. Now, coming on to ERBB4 receptors themselves, they may be involved in mediating the neuroplasticity triggered by NMDA receptors and to provide both structural and functional regulatory elements for neurotransmission and for NMDA receptors. Both disbinding and neuroreglin impact the formation and function of the postsynaptic density. And finally, DISC1 gene, that is disrupted in schizophrenia 1 gene that makes protein that is involved in neurogenesis, neuronal migration, dendritic organization, and also affects the transport of synaptic vesicles into presynaptic glutamate nerve terminals, and regulates cyclic AMP signaling, which affects the functions of glutamate neurotransmission mediated by metabotropic glutamate receptors. The bottom line is these all affect synapse formation. Also, NMDA receptor numbers by altering NMDA receptors trafficking to the postsynaptic membrane, NMDA receptor tethering within the membrane, and NMDA receptor endocytosis that cycles receptors out of the postsynaptic membrane to remove them. The other risk genes implicated are the genes for DAOA, that is D-amino acid oxidase activator, that codes for a protein that activates the enzyme DAO. As previously stated, DAO degrades the co-transmitter D-serine that acts at glutamate synapses and at NMDA receptor. This in turn would alter glutamate neurotransmission at NMDA receptors. Another gene is RSG4, that is regulator of G protein signaling. It impacts metabotropic glutamate receptor signaling through the G protein coupled signal transduction system. When glutamate synapses are active, their NMDA receptors trigger an electrical phenomena known as long-term potentiation. Now, this LTP leads to structural and functional changes of the synapse that makes neurotransmission more efficient, sometimes called strengthening of the synapses. This includes increasing the number of AMPA receptors. These are important for mediating excitatory neurotransmission. This leads to robust neuroplastic influences. There's an old saying that goes like, nerves that fire together, wire together. If something is wrong with these genes, such a synapse would be weak, theoretically causing insufficient information processing in its circuits. This strengthening or weakening of a glutamate synapse is an example of activity-dependent or use-dependent or experience-dependent regulation of NMDA receptors and functionality at glutamate synapses. This may also explain why the course of schizophrenia is progressive and changes over time for most of the patients, namely from an asymptomatic period to a prodrome, to a first break psychosis with robust treatment responsiveness, to multiple psychotic episodes with declining treatment responsiveness, to a state of pervasive negative and cognitive symptoms without recovery. This continues throughout the life as a sort of ongoing remodeling in response to what experiences the individual has. Another important aspect is whether a given synapse is eliminated or maintained. If critical synapses are not adequately strengthened in schizophrenia, it could lead to their wrongful elimination, causing disconnectivity that disrupts information flow from circuits. Normally, almost half of the brain's synapses are eliminated in adolescence. Competitive elimination of weak but critical synapses during adolescence could even explain why schizophrenia has onset at this time. In adulthood, you may lose or replace about 7% of synapses in your cortex every week. As previously stated, abnormalities in the following genes may lead to lack of critical synapses being strengthened, that is, the disbinding, the neuroglin, and the DISC1. And thus, critical synapses may be mistakenly eliminated during adolescence.